Okay, thank you, Johannes, for the introduction and the chance to talk today. So, as Johannes said, we will talk about fibre now, but this will be a little bit different because we'll be talking about studies in animals, but the results we talk about here will have direct implications for human dietary interventions as well. And as Johannes said, this is the campus out here. A lot of this work has multiple people that have contributed to it. So for many of this, I'm just a messenger. So I really want to illustrate that this is a collaborative effort. So what I'll talk about today will be predominantly about the prebiotic concept. So this idea that you can introduce different elements into your diet, which will be resistant to the host's digestive processes, and will find its way into the distal regions where it will be targeted for specific populations. Of course, the ones we want to have beneficial effect for our own health and nutrition. So this is a concept that's been around for some time. The most common ones that we hear about for prebiotic targets are often the lactobacilli and the bifidobacterium. But more recently, we've also heard about efforts to stimulate butyrate-producing populations, such as Faecalibacterium and Rosaburia. These are two of the more predominantly known butyrate producers in the gut. And the reason why we're interested in these is they're often associated with a healthy gut status and a low disease state. So, and in particular today, you'll hear me talk about these two populations in detail. So here on campus, we are interested in health and nutrition in a variety of different animals. So the work from my group in the past 10 years is focused on all sorts of different fibre-digesting ecosystems, ranging from ruminants and exotic herbivores to some of the more kind of unique mammals that we see, or reptiles as well, that we see in the Nordic countries. Fish, obviously, working in Norway. If you're interested in working on applied animal species, you can't avoid salmon. And today, in particular, I'll be talking about elements that are more closely related to human gut digestion. And even though I'll be talking about pigs, we have a lot of interest in how fibre is digested in the distal gut region of not just animals like production animals, but also in humans. And funnily enough, today I'll talk about how understanding how fibre is digested in a moose can actually benefit how we understand fibre can be degraded in a more human-related context. One of the main reasons for this is the fact that the microbes and the actual mechanisms that they use to digest fibre are the same across all these different systems. So examples you see in a cow or in a moose or in a reindeer, we'll see examples in a pig and a human digestive tract. Okay, so I just want to quickly touch on this paper because this was published a couple of years ago now. But one of the key elements we found here is that when we measured the different types of fibres within the rumen of a moose, so we use a plant polysaccharide microarray to detect different types of plant fibres, and we use NMR to detect the different types of monomeric sugars, which are the building blocks that make up these fibres. So what we saw is that when the moose digests its feed, we see an abundance of mixed leakage glucans, we see celluloses, we see xylins, we see mannans. And when we looked at the corresponding sugar that is connected to this original fibres, for most of them, they matched up. So for high cellulose, we saw high glucose, which is what you would expect. For high xylose, or high xylins, we saw high levels of xylose. But one of the ones that really stood out to us was mannans. We saw really high levels of mannans in the moose diet. And when we looked for the building block sugar that should be highly abundant as well, we could not detect it at all. So this kind of told us that not all fibres are being degraded equally in this system, that some fibres are actually being degraded by different means by the gut microbiota. So I'm just going to skip through to one of the final figures from this paper. And what this figure is trying to show is a metabolic network where we connect different microbial populations to different plant fibres and their different predicted activities. So in these grey boxes here, we have different plant fibres that the moose digest. In these circles, we see different microbial genomes that have been reconstructed using metagenomics, which we've completely annotated for function. And these red lines here correspond to the predicted activity of these different populations using metaproteomics. And one of the striking things for here, of course, in this study was we were interested in understanding why we see high levels of mannan and no mannose sugar, the representative building block sugar. And so we dived into the most prominent mannan degrading population in this system. And when we took a closer look at its genome, we saw it had this polysaccharide utilization locus, this toolkit to degrade mannan polysaccharides. So this is a cross-section of a gram-negative cell. You see your outer membrane here, your inner membrane, your cytoplasmic space down the bottom here. So the way this organism was degrading mannan in the moose gut was its outside enzymes here would recognize the fibre, break it down from its large polysaccharide stature into smaller but still complex oligosaccharides. And these complex oligosaccharides would then be brought inside the cell and degraded into monomeric sugars. So no monomeric sugars were being released from this mechanism into the kind of lumen fluid of the moose. So this kind of told us that this organism was using a selfish mechanism where it's taking big pieces, bringing it inside the cell, and then breaking it down inside the cell. So it's not sharing that sugar with its neighbors. So this was determined using, as I said, metagenomics and metaproteomics, but also biochemistry. So we had to biochemically characterize these enzymes and show that they weren't producing any sugars. So this idea of mannan being selfishly degraded is not new. It's actually been shown in several instances in the human gut. So the first paper that did this was back in 2015 by Harry Gilbert's group at the University of Newcastle. And they showed that Bacteroidetes in the human gut would take complex alpha yeast mannan and use the same kind of selfish mechanism where they would take large complex pieces, break them down into still complex but smaller oligosaccharides, bring those inside the cell and then break them down into monomeric sugars. So there'll be no sharing with their neighbours. This is a completely selfish process. And more recently here at NMBU, we had Sabina Lianti-Larosso and Bjorga Westerang had a nice paper on a gram-positive human gut commensal. This was actually from Rosaburia intestinalis. And they showed in this system that it's also a selfish mechanism where you have complex mannans coming onto the outside of the cell. These are beta mannans in this instance, being broken down into complex oligosaccharides where these are reduced into monomeric sugars within the cell. And the interesting thing about both these systems, this is a gram-negative, this is a gram-positive, is that all the machinery required to do this, so the enzymes, the transport proteins, the transcriptional regulators, these are all confined within one genetic locus. So all these pieces are in one part of the genome, which we refer to as a polysaccharide utilization locus. So if I say PULS, which is the acronym for this term, what I really mean is microbial toolbox that can basically do all the job for the specific fibre being degraded. So this selfish means of mannan degradation and gut ecosystems gave us the idea that it could be a suitable prebiotic. The reason being that if there aren't that many mannan degraders to begin with, the ones that we've characterised so far seem to be selfish, so they're not sharing their sugars with the neighbours. So this is, in a sense, something that we can really target a specific population that has this ability to degrade mannan, and it won't create sugars that will feed into other populations in its surrounds. The mannan substrates require a real specific set of K-zymes, and I'll get into this in the next slide, so that these carbohydrate active enzymes. And luckily for us, several of the more famous butyrate-producing commensal bacteria are believed to degrade mannan. So for our case, Rosaburia and Faecalibacterium are two that we know that like to eat mannan. So these are good targets for a prebiotic kind of concept. Okay, so the idea here was to use highly decorated and complex galactoglucomannan. And so this actually was the brainchild of Bjorga Westerang and Lechek, who was his PhD student here at NMBU. And they came up with the idea that mannan that was actually extracted from spruce wood contained the right kind of structural features that would match Rosaburia intestinalis. And one of the key features of this fibre is not just the sugar patterns, but it's also these acetylations on the outside of the cell, outside of the polysaccharide. So these are kind of protective features that must be removed for an enzyme to come along and cleave this into its monomeric sugars. So they use this extraction process to retrieve high quantities of pure acetylated galactoglucomannan. They incorporated this into pig feed, and we fed this to 4 different groups of pigs here on campus. So we had control groups without any mannan. We had varying inclusion levels. So 1, 2, and 4% of this mannan was included into their feed. And because this fibre had unique structural features which matched Rosaburia intestinalis, and this is work we knew from the previous paper I talked about, we actually went into the distal gut of these animals and checked for the activity of Rosaburia in particular. So this was the design of our trial. As I said, we had 4 different groups. We fed them different levels of mannan. We monitored their fecal microbiome over a 4-week period. And at the end of the period, we also used the entire gastrointestinal tract, and we used multiomics to analyse the changes in the animal's microbiome structure. So we use combinations of 16S, metagenome, assembled genomes, and metaproteomics to monitor their activity. So this is the first part. This is looking at the fecal microbiome and how it changed over time in response to these diets. And for this, I'm using 16S analysis, and we're looking at the change of the fecal gut microbiome in response to either a control diet here in black or a green dot here representing 4% mannan. So we can see here at the initial stages, this is the control actually, so we would expect to see no changes between the 2 animal groups being fed this diet. As we change this over time, we can see after 1 week, we didn't see any changes in the fecal gut microbiome. 2 weeks, we saw no changes. 3 weeks, we begin to see a change. And after 4 weeks, this change is quite prominent. That the animals being fed this 4% acetylated galactoglucomannan were giving rise to a microbiome that was structurally different from that in the control animals. So this is looking at 16S analysis, looking at the total microbiome. We also looked at the different regions of the gastrointestinal tract. So we monitored the change of in these different regions depending on the levels of mannan. So again, using the same color scheme where black represents control and green the mannan, and the brighter the green, the more mannan that was being added. And the key thing to see here is that in the upper regions, in the small intestines, we saw no changes in response to this diet. But as we move down into the distal tract, where we would expect fibre-degrading microbes to live, we saw changes. So in the cecum and in the colon, we saw big changes in response to this mannan being added. And when we looked at individual populations, so this again is using 16S, and this was to monitor the changes in Rosaburia populations. And this result you can see here actually is extremely negative, and at the time, extremely depressing. So what this actually was showing us is that when we increased mannan, we actually saw a decrease in Rosaburia populations. So this is the complete opposite of what we wanted to see from the beginning. And this is using 16S, and I'm going to show in a few slides from now why this isn't super depressing as it looks. If you look at Faecalibacterium, and this is another population we suspected of being able to degrade this fibre, we saw the opposite. We saw the result we wanted to see. We saw as we increased the levels of mannan, we saw an increase in the Faecalibacterium and the distal regions, so in the cecum and then the colon. So these are the 2 populations that we're particularly interested in. If we look at some of the other populations, we also saw an effect. So for Prevotella, which is a fibre-degrading genus that's quite abundant in the distal regions, we saw a large increase in response to mannan being added. Catanibacterium, Ealista, and Megasphaera were 3 others that we really saw big shifts in response to this mannan being added. But again, this is 16S, and we're really interested as to the functional responses, which genes are being activated, what is the metabolic responses in these different populations. So for this, we used metagenome assembled genomes, so reconstructed genomes from metagenomes. And what this tree is trying to illustrate is 355 genomes that we reconstructed from the pig gut microbiome. On the outside here, we have mapped proteomes for each of these populations. So we generated metaproteomes for these different samples, and we mapped all these proteins back against the individual populations from which they originated. So each one of these spikes here represents a proteome that's originating from this population. And we did this for both control and mannan fed animals. So what I'm going to do now is zoom in on some of the populations we were specifically interested in. The first one is Rosaburia. So this is this section here in the tree. So if we zoom in on Rosaburia, we saw about 15 different genomes that reconstructed to this genus. We looked at their proteomes, And again, this showed a really negative result. We saw that the Roseburia populations were actually producing more protein in the control and less protein in the mannan-fed animals. And this was the case for nearly all of them, except for one in particular. And this is the one I really want to talk about. So we saw for one Roseburia population, we saw exactly what we wanted to see. We saw a big spike in its proteome, and in particular in animals being fed 4% mannan. So we took a closer look at this genome, this reconstructed genome and its proteome. And the key thing we saw here was it encoded this toolkit that was specifically targeted for the galactoglycomannan. So this fiber that we added that we knew from Roseburia intestinalis was highly specific for this fiber. When we looked at this reconstructed population, we saw this exact same locus within this genome. The other cool thing that we saw was when we looked at the proteome, so if we look at the control and the mannan, we saw that this locus was switched off in the control feed for the most part. And when we fed mannan to these animals, this locus was being completely switched on. So this is the result we wanted to see. We took a look at the genes and compared them against this nice study that Sabina did earlier on the human gut Roseburia, and we saw high synteny across the whole locus. So high gene similarity, high protein similarity across this whole system. So this was for Roseburia, but of course we have 355 other populations in this system. We took a look at some selected others, and I'll go through those in particular. One I really want to highlight is, I'll come back to this. One I really want to highlight is not just the Roseburia, which is what I've talked about here, but it's the Faecalibacterium. So we talked about in the 16S analysis, we saw Faecalibacterium responding positively to this fiber. We took a look at the genome, which corresponded to Faecalibacterium, and we saw a version of the same locus within its genome, this mannan-specific locus. And again, we saw it completely switched off in the control and active in animals that were being fed this fiber. The key thing here that I forgot to mention in the Roseburia slide is we saw these esterases that were specific for removing these acetylation patterns on the outside of this fiber. So this structure that makes it really hard to degrade, we see these special esterases encoded within these populations. So as I mentioned, in the 16S analysis, we also saw a lot of effects in a lot of other populations. And we're trying to understand what this means. Prevotella, we saw a big increase in populations. When we looked through the Prevotella genomes, we saw an increase in their proteome in response to this diet, but not necessarily targeted towards mannan degradation. This was towards short-chain fatty acid synthesis and also fibers that were found in other components of the diet. So these diets contain basal ingredients such as oats and wheat, and which are packed full of different fibers. And we saw the capabilities to degrade those fibers within these other populations being stimulated, especially in animals that were being fed this mannan. So there's this kind of weird effect we're seeing where populations that aren't targeted towards these fibers are actually being stimulated irrespective of their own metabolic capabilities. We also saw the same for lactate-producing organisms, the lactobacillus. Again, these aren't targeting mannan fibers, but we saw if you look at 16S, we actually saw a decrease in their abundance, but we saw a flatlining of their metabolic function, so their activity was maintained irrespective. We saw other populations that were really spiking in the mannan diet, but again, completely unassociated with this fiber. We believe this megasphaera, which produces butyrate, is actually connected to the activity of the lactate-producing populations in this system. And the last thing I just want to point out, I'm not going to go into detail here, is we saw a lot of cross-feeding going on in other populations that had a huge response metabolically to this fiber being added, but from what we can see have no real connections to this fiber metabolically. So we don't know why they're being stimulated, but we're seeing this kind of ripple effect occurring from its addition. So the last thing I just want to add is that why we did this was to try and stimulate butyrate production. And if we actually look at butyrate levels in these animals, we saw an increase. So it seemingly is having an effect. And this is just the last slide I have here, just to kind of summarize this whole microbiota-directed fiber concept. So can this work? We believe it can, but we believe there's three key elements that you have to understand. And one is you have to understand the microbe and its tools. Two is you really have to understand the substrate. And three is even if you get this to work and you hit your bug right on the head, you're still going to have a ripple effect that's going to affect all these other populations, and we really don't understand what that is yet. So this is something that we really need to go forward and work on together. And so I've used up all my time. I just want to finish with acknowledgements. All these people here, hopefully I acknowledged throughout the talk. The ones in black in particular were involved with the work I presented today. So thanks for your time.
Thank you, Philip, for very, very nice and ambitious work and really impressive results. So just a very basic question. Do you plan to take this model into humans, and do you have any plans for that? And also one question from the audience here. Do we need more prebiotics like inulin-resistant starches, galacto-oligosaccharides and others that could modulate the microbiome?
So in response to your first question, we would love to. We would love to do this, collaborate with some people who are doing this in a human setting. So, and I know particularly Sabina is out there listening today, and she's particularly excited in where this goes. In response to the second question, I would not dare to answer that because I honestly don't know enough about those fibres and their effect. So I know there's a lot of connections with the resistant starch and particular mechanisms, but for those other substrates, I'm not brave enough to go there.
So another question, what are the dietary sources of manans? I mean, you talk about trees and you have yeast, and what are the sources?
So legumes are packed full of beta-mannans. Actually, coffee is a good source for beta-mannans. I think there's other vegetables, specific ones, tomatoes, I have a feeling. I don't know. I'm sure Sabina's out there, she'd be listing them all off. But I know especially beans and legumes are a good source for beta-mannans.
Thanks a lot. Any more questions from the web, Johannes?
One more question. Why didn't you see an expansion of the bugs that could use synthetic fibre as a substrate?
Yeah, so that was a good question, and something I just didn't have the time to go into. And I forgot one key message here. The ones that didn't expand, they didn't have this locus. So the one population out of the 15 Roseberry that we recovered, the one that had the locus saw an expansion. The rest that didn't have that locus actually died away. So we think it's really this locus that's the key to being able to degrade this fibre.